Welcome back, ladies and gentlemen. So we are going to continue our discussion today on this idea of just differential gene expression by really talking about this idea of pattern formation. So when we look at the development of multicellular organisms, there's definitely spatial organization in terms of where your tissues belong, where your organs belong. Where's your head? Where's your feet? Where are your limbs? Where's your thorax or your abdomen? And something like these guys. So it really sort of sets up that anterior, posterior, left, right, dorsal, ventral, all of those body axes that if you've ever taken an anatomy and physiology course, that's probably one of the first things you learn. So all of that directionality comes from differential gene expression in really early cells as that embryo initially um, starts developing. So they're molecular cues that control all of that formation and tell your body where they belong, and then start potentially influencing neighboring cells to make sure that their patterns are set up correctly. So when we look at this pattern formation, it's been incredibly studied, very extensively studied in fruit flies or Drosophila. And what we see here is a great example of some more of that cytoplasmic determinant. So when we look at where those certain cytoplasmic determinant molecules are sort of isolated in that unfertilized egg, it already is setting up the axes before fertilization even happens, so that once that fertilization occurs and those cells start going through cell division, certain concentrations of those cytoplasmic determinants are set up to be isolated in the head for these sort of quote unquote head cytoplasmic determinants. The cytoplasmic determinants that are meant to be in the thorax end up in another subset of cells, and then the abdomen towards the back as well. And so this is going to combine all sorts of information from a lot of different fields, anatomy, genetics, biochemistry, to all sort of end up discovering the fact that not only do these principles exist in these fruit flies, but that they're common to incredibly large numbers of species, including humans. So, to the point that these two scientists won the Nobel Prize in 1995. So they demonstrated that these genes directly impacted the developmental process, specifically these things called homeotics or Hox genes. That's a subset of genes that exist in varying numbers in terms of the number of copies that exist in certain species, um, switches of when they get turned on and get turned off across developmental stages of those early embryos, but ultimately they exist to control pattern formation in the embryo, in the case of the fruit fly, the larva, and the adult. And we know that these exist in a large number of organisms outside of these fruit flies that they were studying. But what they did was they were actually able to go in and create what they call gene knockout studies. And what that really means is they go in and purposefully mutate some of these Hox genes to a point of either turning them on, turning them off at the wrong times, completely knocking out their ability to function at all. And what they did was then look at the resulting offspring and what sort of weird mutations did occur. And so here you have a normal wild type fly with their cute little antennas and their little bug eggs, right? And here's a particular example of one of those mutants where they ended up messing with the coordination of when those Hox genes got turned on and turned off in that development. And instead of antenna, they got legs growing out of its face. They were able to create mutant flies that had two heads on either end, who had no heads at all, who had more legs than they were supposed to, so on and so forth. So, really sort of delving into mutating one gene at a time and seeing what it resulted in was able to give them the understanding of how these Hox genes have such a vital role in the formation of these body plan components. So the other thing that they also sort of inadvertently understood is by creating these mutants, by doing selective breeding experiments, they also noticed that some of this got complicated by this idea of an embryonic lethal. And what that means is that when they were looking at this normal segmentation and trying to mutate these genes, occasionally they would get numbers that didn't seem to add up statistically. Um, and what they eventually were able to backtrack is that some of those mutations were just so out there, so devastating to the formation of those body plans that the embryo just never developed. So that is exactly what it sounds like. So they have these lethal mutations that are so devastating that the embryo can't function, and so there never is a baby fly as a result of that. Collectively, though, they ended up finding that there are 120 genes essential for normal segmentation in these fruit flies, and we see similar patterns again across a lot of organisms that have this very distinct body plan in place, humans included, which is pretty crazy. They are very, what we call highly conserved genes, meaning that their evolution is probably relatively old because we see it across so many different um, types of animals that exist. Something else that we have discovered that is sort of another idea of these cytoplasmic determinants are these things called maternal effects genes. And these are genes that encode for those cytoplasmic determinants specifically in establishing those axes of those bodies of those Drosophila. So you may also come across them as egg polarity genes. It's a similar idea. It's another similar set of genes in the sense that it's going to help set up body plans and pattern formation in those body plans. But here, these guys are specifically going to set up anterior, posterior axes of those embryos, as well as dorsal ventral. So they control the orientation of the egg and consequently the fly itself. So when a mutation happens in the mother itself, it results in a mutated phenotype in the offspring. And it doesn't really matter what the offspring's actual genotype is. So if you remember, back this up a step, we talk about genotypes and genetics, right? So when you're doing those traditional Punnett squares, you're used to writing genotypes as a capital A, lowercase a, or two capital A's or two lowercase a's, right? Are we homozygous dominant? Are we heterozygous, homozygous recessive? And it was typically when we would talk about that, the interplay between the allele, right, one big A or little a, that mom gave you, as well as the allele, the big A or the little a that dad gave you. So if you were homozygous dominant, you got the same copy from mom and dad, and so you had the dominant phenotype, right, the physical expression of that trait. If you got a lowercase from both parents, you had the recessive, and if you had a uppercase from one parent, a lowercase from the other, then you were heterozygote and you would probably show the dominant trait if it was a traditional Mendelian um, characteristic. But in this case, when we look at maternal effects genes, it doesn't matter what dad gave you. It's all going to be because these cytoplasmic determinants are already set up in that egg before dad ever drops off his genetic information. So if mom has a mutated maternal effect gene, then there's going to be a mutation in the offspring. And this really comes down to the fact that those mRNAs, those protein products, these are those cytoplasmic determinants, are products of the gene that are placed in the egg while they're still in the ovary. So the mutation is defective in that product, and it's that product that ends up determining these axial directions, not a result of what happens once dad's genetic information gets dropped off and then we express that information. So hopefully that makes sense. So this is set up pre-fertilization, and so sort of, if you want to put it this way, the damage is already done. It's not going to matter what dad's genetic information comes and brings in because the molecules are already produced pre-fertilization. So the bicoid gene is a good example of a maternal effect gene in the sense that this particular gene sets up a concentration of mRNA molecules, bicoid mRNAs, and they are there to distinguish head from tail. So what you should see is a high concentration, I should say, of that mRNA in an unfertilized egg that then quickly goes through protein synthesis and creates that bicoid protein in that very early embryo. Doesn't matter what dad dropped off because the mRNA is already in existence. And so you should have these high concentrations here so that that sets up your head, and then it trails off. It's a concentration gradient, it trails off, and the least concentrated ends up being the tail. So if there is a situation where there is mutated bicoid genes in the mother, then we may end up in a situation like this, where there is a weird concentration gradient that happens here. There's no head, but two tails. Or it could be the reverse, two heads and no tails. Um, so that ends up duplicating posterior structures at both ends or anterior structures at both ends by messing with the resulting concentrations of those mRNA molecules. So it should be in this high concentration at the head, lowest concentration at the back end at the tail, um, in order for that bicoid gene to do its job correctly and set up the head versus tail axes in that offspring. So it's an example of what we talk about as a gradient hypothesis, so that these gradients are substances called morphogens properly establish that embryos' axes as well as other features that would come along with that. So number of legs might be a result of having certain concentrations of something like this bicoid gene as just an example, bicoid proteins, to set up the proper number of legs, that the legs are in the right place, for example. Um, so that's the idea of these morphogens. So what this research ended up giving us above and beyond is just, hey, these fruit flies have a head and a tail, and this concentration gradient is a result of that. It helped to really identify specific proteins that are required for some of those very early steps in that pattern formation. Helped us to understand that the mother's role in that early embryonic development is incredibly important, even more so sometimes than the interplay between the genes between mom and dad. And just understanding those key developmental principles in the sense that these molecules create this gradient. And that really turns into uh, influencing the genes in those cells to be able to then continually differentiate their own gene expression to get the proper positioning, to get the proper organs in the right place, limbs in the right place, so on and so forth.
Once we go from actually differentiating our cells into what they're going to become and starting to, get their morphogenesis, starting to become their cells, we get pattern formation. And they're a set of genes called positional genes, and they set up the body axis. What I mean by body axis is they tell the developing embryo where front and back, right and left, up and down is. Now, the picture I've got on the right is of a Drosophila embryo, a fruit fly embryo. And the reason we got that up is when it comes to genes helping to form body plan is one of the best studied and best understood organism. So you can see that there's all those little like fluorescent bands across the larva or the embryo. Those are different genes that have been labeled by researchers, and as those genes are expressed, they take up different positions on the body. So know that pattern formation is setting up right, left, front, back, up, down, all that good stuff, and positional genes are responsible for doing that. More specifically, there's a set of genes called homeotic genes, and I'm going to put this down real quick because you'll see it a lot. Homeotic genes are also known as Hox genes, and Hox genes are responsible for setting up like body segmentation. So our pattern genes set up front, back, right, left, up, down. Homeotic genes determine, all right, here's where here's where ears are going to be, neck, chest, arms, legs, etc. Interesting thing is, homeotic genes are pretty consistent from one organism to the next. So right here, they show you a human chromosome, here's a fly chromosome, and they have color-coded all of the different homeotic genes. And you can see in the fruit fly, the red homeotic gene tells where the proboscis is going to be. In humans, it tells where the ears are going to be. Now, there's one gene for that in a fruit fly. There are 3 genes for that in humans. And then as you look down the rest of this thing, like the blue genes right here, signal for where the wings and the thorax are going to be. The thorax is like the chest of the fruit fly, and it shows where the chest is going to be in humans. So as our embryo is developing, these homeotic genes are turning on and saying, all right, this part of the embryo is going to become a chest, this will become an ear, this will become a leg, this will become like a face. And it's kind of interesting to think, evolutionarily speaking, that the same genes are present in most other organisms that have got a pretty set and determined body plan.